Hello everyone, today I will introduce the article Machine Learning Predictive Modeling in Nanomedicine-Based Cancer Drug Delivery co-authored by Rohan Sahu, Sanchit Arora, Dinesh Kumar, and Ashish Kumar Agrawal. Now, let's officially begin the introduction. This article systematically elaborates on the pivotal role of machine learning in nano-drug development, provides an overview of commonly used ML models including logistic regression, decision tree, random forest, gradient boosting, and k-nearest neighbors, presents case studies demonstrating the practical application of ML in nanoformulations, and discusses the challenges and future prospects of this field. The application of machine learning in nanomedicine typically follows a standardized four-step workflow. Firstly, data collection and cleaning are performed, which involves acquiring drug molecular property parameters and formulation process parameters, followed by the removal of noise and outliers to establish a foundation for modeling. Subsequently, model construction and validation are carried out, requiring the selection of an appropriate machine learning model based on specific research objectives, along with a reasonable division of training, validation, and test sets to ensure the model's generalizability. Then, feature importance analysis is conducted to identify the variables that have the most significant influence on key prediction targets, thereby providing clear direction for subsequent experimental optimization. Finally, by systematically comparing the performance metrics of different models, the optimal model best suited for the specific task is selected. Core highlights of this research are reflected in two key aspects. First, the proposal of a standardized machine learning workflow that fully integrates ML into the entire nanodrug (RUD) pipeline from data cleaning and model training to result interpretation, forming a closed-loop workflow. This workflow has revolutionized the traditional trial and error (RUD) model and realized predictive (RUD). Second, the focus on bottlenecks and pathways for clinical translation, which clearly identifies insufficient model interpretability and lack of clinical validation as the core obstacles hindering the clinical application of ML-driven nanomedicines. Practical solutions are provided from perspectives such as technical optimization and regulatory considerations. In this section, we will present several case studies. Case 1, ANN optimization of silver 5-Fu zeolite-based nano-drug delivery system. An artificial neural network was employed to optimize the zeolite-based nano-drug delivery system loaded with silver and 5-fluorouracil. Through modeling and analysis of training and validation data, the model accurately predicts the cytotoxic effects under different drug ratios, providing an optimal scheme for the design of nano-drug delivery systems for melanoma treatment. Case 2, Light GBM for predicting nanocrystal particle size and PDI based on 910 sets of nanocrystal preparation data. A predictive model was constructed using the Light GBM algorithm, successfully achieving accurate prediction of nanocrystal particle size and polydispersity index PDI under different preparation processes. Experimental validation results show that the predictions are highly consistent with experimental values, significantly reducing the experimental workload of process optimization. Case 3, ANN model for predicting nanoparticle cellular endocytosis behavior. Breast cancer diagnosis. An artificial neural network model was established, taking the surface chemical properties, particle size, zeta potential, and other characteristics of nanoparticles as input parameters, and endocytosis efficiency as the output index, to predict the endocytosis behavior of breast cancer cells. This model can not only distinguish cancer cells from normal cells, but also identify cancer types and disease stages, providing a new tool for the precise diagnosis of breast cancer. This study outlines a complete machine learning workflow for nanomedicine development, from data processing to model interpretation, providing a clear path from trial and error to rational design. It lowers the technical barrier for researchers by demonstrating, through practical examples, how machine learning can optimize formulations and predict outcomes. The work integrates with existing computational tools, emphasizing data standardization and model interpretability to ensure predictions can effectively accelerate real-world drug development.